Hey everybody, welcome back to General Biochemistry Lecture. We're on the final chapter of the book, which is chapter 28. We're bringing everything together and talking about the regulation of gene expression. This chapter is going to be broken up into 2 videos, and we're only covering sections 1 and 3. If you're interested more in bacteria, read section 2, but we are not going to cover it in detail. Some bits and pieces, we're going to talk about a little bit, but by and large, we're going to try to focus on humans since we're all kind of interested in medicine and health and things of that nature. Here we go. These are your key principles. Always try to go back to these and think through examples that kind of exemplify these different statements. Make sure that you understand what they mean. So for example, when we're talking about regulation is expensive, by the end of this chapter, you should be able to talk about why it's expensive. So there are 7 processes that affect steady state concentration of a protein. And we've talked about all of them in varying degrees of detail. So there's the transcription initiation level where, you have to make some kind of an mRNA transcript in order to make a protein. So there's regulation there. Then there's regulation at the post-transcriptional, level where you process the messenger RNA, remove introns, all sorts of stuff, splicing. Then there's RNA stability. So we said that messenger RNA, there's lots of endonucleases and exonucleases that want to chew that transcript up. There are things in place within the molecule itself and the structure that prevent this from happening or at least slow it down, but there are still things internally about the messenger RNA that can make it more susceptible to degradation. Then there's translational regulation. So does a ribosome bind to it? How quickly do ribosomes zip through and make a protein? All that kind of thing. Protein modification. So once you have this nascent polypeptide, there are things that need to happen to make sure that it takes on the right fold. There may be some post-translational modifications like phosphorylation, prenylation, glycosylation. All of those things take time. They have to be trafficked to certain places. And then, of course, that leads us to protein transport. That is too, maybe it needs to get glycosylated in the ER. Maybe it needs to get sent to the membrane so that it can find its buddy and pair up with its protein-protein binding partner. And then, of course, there's always protein degradation. So maybe something gets ubiquitinated and it's tagged for protein degradation. That is kind of the battle of a protein, So all of these things affect the concentration of any given protein in the cell. As you can see, regulation is pretty complex, but it gives the ability to fine-tune the amount of protein that you need available very quickly and in many different ways. So as we talk about the regulation of genes, I want you to try to zoom out, particularly for the last section of class, starting with looking at DNA metabolism, and see how all the things that we talk about fit into the various topics we've covered with DNA, RNA, metabolism, protein metabolism, and then thinking about some of the experimental procedures we covered in chapter 9. So that's just a heads up for how to help organize this information. And I'll give you more detail about that as we go on. So first we're going to talk about proteins and the RNAs that are involved in gene regulation. Now there are lots. So we'll talk about some kind of general classes. We'll talk about some protein motifs. We'll talk about some examples. But what I really want you to do is look at those examples and see how they work. And then think about how you can, interrogate those. So how can you learn more about them? What techniques can you use from chapter 9? And how can you take what you understand about one system and apply it to another? So we're going to start off with talking about RNA polymerase binding to DNA at promoters. So remember that RNA polymerases make messenger RNA. They sit down on a promoter along with a bunch of other factors that help to guide the polymerase to the promoter and to keep it stuck to the promoter and enable promoter clearance so that you can make that messenger RNA. Promoters are the sites on the DNA template that are generally found near points where RNA synthesis actually begins. The regulation of transcription initiation changes how the polymerase interacts with the promoter. So we talked about this in chapter 26, like, it just happens and, there's no regulation, but there's a lot of regulation. The differences in promoter sequence affect the polymerase binding and the frequency of transcription initiation. So this is just one level of regulation. If you don't have a lot of RNA polymerases sitting down on those promoters, then you're not going to have much of the mRNA transcript, which means you're not going to have protein. Remember that there are more things than just messenger RNAs that are made. So there are lots of non-coding RNAs that are essential for regulation processes, and these are also regulated in a similar way. In E. coli, most of their promoters have a sequence that's close to a consensus sequence, and their mutations away from the consensus sequence usually will reduce the amount that the promoter gets bound by RNA polymerase, so it's not as functional. You'll still make some messenger RNA, or whatever RNA, but it will usually decrease the promoter function. And the closer you are to that consensus sequence, the more enhanced promoter function will be. So down below, there's a piece of DNA. We've got some elements here. The up element, which is kind of like an enhancer region. Then you've got the -35 region and the -10 region. And these are the two regions where you're going to see that consensus sequence that's going to help either promote or prevent the RNA polymerase from binding well and starting the transcription process. Now, there's a lot of different genes, right? Some of them are going to be transcribed more readily, others less frequently. There are what's called housekeeping genes, and these are genes for products that are required at all times and they're expressed continuously. So some examples would be enzymes of central metabolic pathways like glycolysis. That's always going on pretty much. Even if it's at a very low level, you still need those enzymes. The cellular concentrations of the proteins encoded are going to vary, but you're always pretty much going to have those promoters be busy. So you're going to have RNA polymerase sitting down, making some mRNA transcripts. Then there's constitutive gene expression, and that means that you have expression of a gene at approximately the same level, always and forever. The RNA polymerase promoter interaction influences the rate of transcription initiation. So again, this is just one level of regulation. So transcription initiation is also regulated by proteins and RNAs outside of the RNA polymerase. Some of these are specificity factors, which we talked about the specificity factor with bacteria when we covered RNA metabolism. And there's the sigma 70 that is responsible for guiding the RNA polymerase to the promoter in bacteria. There are other sigma factors, but sigma 70 is responsible for the majority of the work in bacteria. However, those other specificity factors can guide the RNA polymerase to other promoters to induce the formation of those transcripts. There are also repressors, which restrict access of the polymerase to the promoter, and activators, which activate or enhance the polymerase promoter interaction. To add to the complication, RNAs themselves can participate in regulating RNA polymerases. So there are non-coding RNAs, which are RNA molecules that are not translated. So you make a transcript, there's RNA, but you don't take that RNA and make protein. The majority of the RNA that gets made is in this category. This is a big catch-all category. Within that, there are these long non-coding RNAs. So let's do a little bit of a diagram. So we have non-coding RNAs. Within that, we've got several different types. The long non-coding RNAs, we could have, for example, tRNAs. You remember those guys? There's ribosomal RNAs. So these are all non-coding RNAs, and we're going to add to this list. But I just want to make sure that we get that terminology right. All of these RNAs can be considered non-coding RNAs as long as they don't get translated to protein by a ribosome. The long non-coding RNAs are non-coding RNAs that are more than 200 nucleotides long, and they don't have an open reading frame. So it's a long strand of RNA that doesn't code for anything. But more and more, definitely over the last decade or two, researchers are finding that RNAs have a lot of roles in regulation in the cell. So most of the transcripts that are made are for regulation purposes, and that the messenger RNAs are just a tiny fraction of what RNA does. Here are some of the known functions of the long non-coding RNAs. So regulation of nucleosome positioning and chromatin structure, control of DNA methylation, post-transcriptional histone modifications, which we're going to talk about chromatin and histones in a little bit as a brief aside because we did not cover chapter 24. But if you're interested in chromosomes, you should definitely check out that chapter. There's other things here too about the functions of long non-coding RNAs. Be familiar with them, but the real idea is to understand the potential for what they do, so when we look at case studies, you'll be able to understand. So let's go back around to those bacterial specificity factors. So a specificity factor is an amino acid sequence that mediates promoter recognition and binding. So the sigma subunit, like we said, of the E. coli RNA polymerase, that's a specificity factor. Sigma 70 helps the polymerase recognize the majority of the promoters in E. coli. However, if you have heat stress, sigma 70 is replaced by sigma 32. And the numbers just tell you how large the sigma factor is, so its weight in kilodaltons. When sigma 32 is under control, it's going to direct that RNA polymerase to special promoters with a different consensus sequence. And this is going to cause the E. coli to express genes that are involved in reacting to the stress of heat, the heat shock response. Those proteins, it makes sense that you don't want to have those around all the time because you're not always under heat stress. So why go through the trouble of making all those proteins when they're not going to do anything? Remember, protein manufacturing is expensive. So you don't want to do it unless you have to. So again, to put this into perspective about what it looks like, this is an example of a sigma 32 promoter region. Notice that these two purple regions are very different. Only the heat shock response will get that sigma 32 to be expressed and then bring RNA polymerase to these specific promoters to turn on the proteins that will help maintain the E. coli in this heat stress until it's gone. Now there are some eukaryotic specificity factors. We don't really think about them in that way, like bacteria, But some of the general transcription factors can be considered specificity factors. And one example of that would be the TATA binding protein, TBP. Bacteria also use repressors. And you'll see that we use very similar mechanisms, but just the chemistry looks different. So repressors are proteins that block RNA polymerase binding, or they prevent the polymerase from moving along the DNA. But the result is the same, that you cannot get proper initiation or elongation to create an mRNA transcript. There are operators, and these are binding sites on the DNA that bind repressors. They're usually located near the promoter. They can be a little bit upstream, a little bit downstream, but they're usually close to the promoter. Then there are effectors, and these are small molecules or potentially even proteins that regulate repressor binding to DNA to alter transcription. So these molecules will bind to the repressor and cause some kind of conformational change, and that can either cause them to dissociate from the promoter and allow promoter clearance, or any number of things. We're going to talk about some examples. We're going to nail down some more vocabulary first with some generic scenarios, and then we'll talk about a real bacterial operon. So we've got negative regulation, and that means that you've got a repressor that blocks transcription. So negative regulation, think repressor. There are two different scenarios that you can envision with negative regulation. The first is that you have a repressor protein that is sitting down on the promoter, and this promoter is off. You're not making any mRNA transcript. However, a molecular signal comes along, and that's this green diamond, and it binds to the repressor, and that causes the repressor to change its shape and it suddenly doesn't want to bind the promoter region anymore. Now, that repressor is gone, and the RNA polymerase can bind, and it can create that messenger RNA transcript. So that's one example of negative regulation. The other example would be, we start with a gene that's on. Okay, there's nothing obstructing its promoter region. RNA polymerase can sit down and make that mRNA transcript all day long. But along comes a molecular signal that binds to a repressor, and then all of a sudden you have this repressor that's bound to the promoter. Now the gene is off. You're not making any more messenger RNA. These are both examples of negative regulation because of the presence of a repressor. Whether you start with the repressor on the promoter or the repressor is recruited to the promoter, doesn't matter. They're both examples of negative regulation. Now let's talk about eukaryotic repressors. So gene regulation by a repressor is less common. You'll see it more often in lower eukaryotes like yeast. The binding site for a repressor may actually be really far from the promoter in eukaryotes. Now let's talk about the flip side, activators. We'll start with bacteria first, as usual. Activators are proteins that enhance RNA polymerase promoter interactions. Their binding sites are often adjacent to promoters that are very weakly bound by RNA polymerase, or it won't bind RNA polymerase at all on its own. So these promoters need a little bit of help, and they get that help from an activator. Whenever you see activators used, we're talking about positive regulation. So again, regulation by means of an activator protein that induces transcription. Activator, positive regulation. Now we'll go through two scenarios of positive regulation. So let's say that there's RNA polymerase and you've got your activator present on its binding site. That means that this gene is on. You've got your activator, you've got your RNA polymerase, you're making messenger RNA. We have to have a way of turning this gene off because it may not be something that we want to continually express. In comes a molecular signal, binds to the activator releases the promoter region or its activator binding site. And now this RNA polymerase is no longer stable. It can't form a stable pre-initiation complex to actually go through and make messenger RNA. So that it's going to fall off. And that gene is turned off because that RNA polymerase can't bind the promoter very well. The other example would be the reverse, where you're starting with a gene that's off. We have very weak binding of RNA polymerase or none at all. Then there's a signal that causes an activator to bind the binding site right next to the promoter, and it will recruit the RNA polymerase, help it to stay bound, and form that initiation complex so that it can make a messenger RNA. That means that the gene is turned on. Now we're using off and on as binary signals, but the truth is that there are many, many shades of gray in between. Off may be really 100% off, but on is quite variable. So eukaryotic activators are used very widely. This is quite common. And enhancers are DNA sites that bind activators that are really distant from their promoter. So just like the activator binding site in E. coli, how those are usually right next to the promoter, in eukaryotes, those sites are enhancers and they're kind of far away from the promoter. Activator binding can affect the rate of transcription at promoters even though they're very far away. And understanding a little bit about chromatin structure will help you understand how that can work. So let's talk about some more players, because clearly with eukaryotes, because we have all this stuff happening upstream of the promoter, there's got to be a way for them to come together. Architectural regulators. These are proteins that bind to sites between a promoter and the activator or repressor, and they facilitate DNA looping. So they change the structure of the DNA, and it brings sites together that normally wouldn't be together. There's also co-activators, which are proteins that mediate the interaction between activators and RNA polymerase. So as you can see, the plot thickens. Protein repressors may bind in their place instead to make it so that you can't have that interaction between RNA polymerase and activators. So in our kind of generic scheme here, we can have an activator sitting down on its enhancer site. Then there's some architectural regulatory binding sites in brown. And then you have your promoter. If you have those architectural regulator proteins come in and bind, then you're going to change the structure of the DNA. And it's the local structure. So that you're bringing two pieces closer together. Once this happens, then you can imagine that when POL2 comes along with a co-activator, the co-activator is like the bridge. It will have one area where it makes contact with the activator and another area where it makes contact with the RNA polymerase. And in between is just kind of the stuff to keep those things together, because those two domains on their own may be too small. They may not be stable. So you have some more, protein fluff in the middle so that you get the spacing right. Now think about what it would be like if instead you had some kind of a repressor coming into the mix. So maybe you have the DNA changing its structure, but you don't have a coactivator here. Maybe you have a repressor that's either bound here or something that is on the coactivator and something else needs to bind to relieve the repression. So there are lots of different complicated schemes you can imagine, that would allow modulation of all these events. So many bacterial genes are actually clustered and regulated in what's called an operon. And an operon is just a gene cluster along with the promoter and any additional sequences that allow the whole thing to function and be regulated. Many of the genes in the same operon encode subunits of large protein complexes, which makes sense, or they're involved in related processes, and you want to be able to have all of those things, regulated in the same way. It just makes sense. So let's say, for example, our good friend glycolysis, lots of enzymes involved. It would make sense to put them all under the regulation of the same operon, because then when you want to ramp up glycolysis, you turn on one promoter, and then you have all these mRNA transcripts, you make all this protein, and there you go. Versus having to turn on an individual promoter for each individual gene. That's a lot more expensive. Down below is an example of an operon. So you've got some kind of an activator binding site that's upstream of the promoter. Then there's the promoter, and then there's what's called an operator, and that's just a repressor binding site. Then after that, you've got all of your genes that are going to be transcribed as a unit. In bacteria, you'll often see polycistronic messenger RNAs, which means that it's just one long strand, and then you make a bunch of protein. So we're going to talk about lactose metabolism in E. coli, and we're going to talk about the lac operon. This is covered in more detail in the second section of this chapter. If you're interested in it, by all means, check it out. So lactose can be used as food for E. coli and other bacteria, but we're going to focus on E. coli. Beta-galactosidase is an enzyme that will cleave lactose to make galactose and glucose. There's a channel that is expressed in E. coli that allows for lactose to come into the cell, and that's called galactoside permease. It's a transporter of lactose. Once the lactose comes in, beta-galactosidase can do its thing, and the primary pathway is to hydrolyze these two sugars and create galactose and glucose. However, there's a little bit of isomerization activity that creates this allolactose, which is important. It's a signal molecule. So hold on to that in your back pocket. The lac operon is subject to negative regulation. So automatically in your mind, negative regulation, think repressor. The lac operon has the genes for the beta-galactosidase, the permease, and also a transacetylase that modifies toxic galactosides to aid in removing them from the cell. This operon is repressed when there's no lactose around, but that repression is not absolute. So you still get a little bit of production of all these different enzymes. It's just much, much lower. This figure at the bottom shows you what exactly is in this operon. And we're going to talk about it piece by piece as we go through. So the LAC-I encodes the LAC repressor protein. It has its own promoter, which makes sense. It can't regulate something if it's not expressed. Mutations in this protein result in constitutive synthesis of the operon, which means it's on. It's just always on. The LAC repressor binds to O1, which is the operator for the LAC operon, the main one. O2 and O3 are kind of like the sub operators, and the LAC repressor will bind to either O2 or O3. The LAC repressor actually works as a tetramer, which is kind of crazy. Most of the time you'll see these repressors working as dimers, but hey, there's always room for a little bit of difference. So the LAC repressor binding makes a loop, and that blocks transcription initiation. So when you see the little feet of the LAC repressor, it brings together these pieces of DNA that normally wouldn't be closer together. Now, I hate going backwards in slides, but I think that it is important to do so here. Notice how when we lay everything out in a row, you've got the main operator here, O3, O2. They're not exactly right next to each other. But what's in between them is this promoter region or part of the actual LAC-Z gene, which is LAC-Z encodes for beta-galactosidase. Now we're going to jump forward and look at what it looks like when the DNA is looped. O1 and O3 in this case are close to each other, and that promoter region is looped and inaccessible. The same will happen if O1 and O2 are bound, only instead of it being the promoter region, the gene itself, the LAC-Z gene, is inaccessible. And since all of these genes are transcribed all in a row, if that RNA polymerase can't sit down on the promoter and start making the construct at the beginning, then you're not going to get any of those other genes being made either. You're not going to get any other mRNAs. Allolactose is the inducer of the LAC operon system. So remember how I said it was important, even though it's a very small shunt, that allolactose is an isomer of lactose, and it will bind to the LAC repressor to cause a conformational change, which makes the repressor dissociate. That will turn on the expression of the LAC operon genes. So remember how there's a little bit of basal expression of this whole operator, even though there's a repressor bound? That allows for the detection of lactose entering into the E. coli. Once that's detected via making some of this allolactose, and that can bind to the repressor, now you can upregulate production of all of the things that were in that operon to deal with the fact that you have this source of lactose waiting to be used for energy. In the lab, we actually use something that is non-hydrolyzable, meaning that you can't cut it like you would other galactosides. And we use IPTG, isopropyl thiogalactoside. It will induce the LAC operon, but you can't metabolize it. So here, you can't do anything with that sulfur. So you cannot, you can't cut it, and it will just cause the bacteria to keep chugging along with whatever operon that is inducing. So it's a great tool in the lab because you can induce your cells to grow whatever protein you want whenever you want. So regulatory proteins, we've been talking about them, they have discrete DNA binding domains. And what this means is that they'll bind to a specific DNA sequence. There are some DNA binding domains that have characteristic structural motifs that interact with the DNA. Now remember, think back to when we were talking about DNA and RNA. We've only got 4 bases, and what they do with each other is hydrogen bond. They're hydrogen bond donors and acceptors, and some of those are exposed in the major groove of DNA. And if that sounds really foreign, go back and look at chapter 8, where we talked about the nucleotides and DNA and RNA structure. So the existence of these exposed major grooves in DNA permits discrimination between base pairs. So most of the protein DNA contacts have some kind of specificity with hydrogen bonding to detect different pairs like AT versus GC. So this just gives you an idea of the major groove and the minor groove, which the minor groove is used as, part of recognition, but the major groove gives more. And what we're showing here is just the different places where you have hydrogen bond donors and acceptors. So the blue is an acceptor, the red is a donor, and then the purples are other, which means maybe you can't do any hydrogen bonding there, maybe that hydrogen is bound to a carbon, but you can still detect that difference, and it can be used as recognition. Then the yellows are methyl groups, which can also be used as recognition. So all of these little pieces can be used by a protein to detect certain sequences of DNA. Now there are some interactions between certain amino acid residues in DNA base pairs. Asparagine, glutamine, glutamate, lysine, and arginine side chains are most often hydrogen bonded to DNA bases. But there is no simple amino acid base code to say, asparagine always bonds with AT pairs. Like, that doesn't exist. You can see all different types of modes of interaction with all of these residues. But what I do want you to think about is what characteristics do these side chains have, the amino acid side chains, that allows them to interact with the bases. So think about that. The structures of the amino acids and the structures of the nucleotides are something that should still be floating around in your head. With the nucleotides, you didn't have to draw them, but you should be familiar with their structures. The amino acid residues, you're still responsible for drawing those. So use that as a refresher for exam 4 and for the final. So here's some specific examples of amino acid residue base pair interactions. So glutamine and asparagine can hydrogen bond with the N6 and N7 positions of adenine. So that just means the N, so looking at nitrogens, and then the hydrogens that are attached to them. So the N tells you what the atom is, the 7 tells you the position within the ring. Arginine can form 2 hydrogen bonds with the N7 and O6 of guanine. These are just 2 examples. There are many. Oftentimes, what DNA binding sites look like are their inverted repeats of a short sequence, aka a palindrome. So think about when we were talking about restriction endonucleases in chapter 9. The majority of those recognition sites are also palindromes. So the things that we're talking about here with the DNA binding sites, it applies not just to the regulatory proteins that bind DNA, but also to other proteins that do work with DNA, like exonucleases and endonucleases. Usually 2 subunits, but you could have more than that, of a regulatory protein will bind cooperatively to the sequence. And we saw an example of more than 2 with the lac repressor. There were 4. So it's kind of unique, but hey, that's what science is about. So now we're going to talk about some of the protein motifs that you see most commonly in DNA binding proteins. So oftentimes these things are modular, and there's a DNA binding part, and then there's maybe another part for some other function. If you need help kind of refreshing your brain with some of the terminology that we use to talk about the motifs, go back to chapter 4, I believe, on protein structure. That will be very helpful here. I told you it's full circle. So you pretty much have to flip through the whole book and remember so that this chapter makes sense. The first motif we'll talk about is helix turn helix, abbreviated HTH, oftentimes in papers. It's a small motif, about 20 amino acids, and it's just 2 short alpha helical segments that are 7 to 9 residues long, and they're separated by a beta turn. The lac repressor, its DNA binding motif is a helix turn helix. So what's shown here is the lac repressor. The lac repressor is huge, but only a teeny tiny bit of it is actually bound to the protein. So what's boxed here is the helix turn helix motif that recognizes that that regulatory piece that's within the promoter region. Each one of the subunits of the lac repressor, remember that there's 4 individual molecules in this, it's a tetramer. Each one of them has its own DNA binding site. That's the helix turn helix motif. There are other residues involved in tetramer formation, which are shown here. But these motifs are really, really small, and they would be incredibly unstable if it were just those 2 things. So all this other stuff is mainly just to stabilize the interaction between the different subunits and to solubilize the helix turn helix, tetramer formation, and allow this protein to actually exist. So it's kind of crazy. It seems wasteful, but without all of this extra protein fluff, you wouldn't have regulation because the helix turn helix on its own is not stable. Within the helix turn helix motif, there's what's called the recognition helix. And that's the alpha helical segment that has many of the amino acids that actually interact with the DNA in a sequence-specific way. Emphasis on sequence-specific. You can have other interactions outside of that recognition helix, but that may just be for general DNA binding, more towards the backbone versus the specific sequence of the DNA itself. It protrudes from the protein surface so it can actually access the DNA, and it's positioned in or nearly in the major groove when it's bound to DNA. And remember that major groove has a lot of discriminatory hydrogen bonding and recognition sites, so you can tell the difference between the different DNA sequences. Another motif you'll see very often is called the zinc finger. It's a little bit bigger than a helix turn helix, about 30 amino acid residues, and it's pretty much an elongated loop, and it kind of looks like a finger. And it's held together by a zinc ion. That zinc is coordinated by 4 residues, either 4 cysteines or 2 cysteines and 2 histidines. Imagine what might happen if one of those were replaced with something else that couldn't do the coordination of zinc. What might happen to that motif? Just throwing it out there. The zinc itself is not involved in coordinating the DNA. It is structural. So it is only structural. It's not involved in DNA recognition. Lots and lots of eukaryotic DNA binding proteins have zinc fingers. And remember, you're not necessarily just going to have one of these motifs. You could have many. You could have, 3 or 4 zinc fingers all in a row. And all of those things are specific for, binding and poking at different areas of a particular DNA sequence. So the interaction of a single zinc finger is actually kind of weak. So you usually see zinc fingers in multiples, and that enhances binding dramatically. Some zinc fingers have residues for sequence discrimination, but others will just bind non-specifically. And this is a major difference, not just in the structure, zinc finger versus helix turn helix, but zinc fingers are used in just kind of broad, here's some DNA, I want to bind to it. Zinc fingers can also function as RNA binding motifs, which is another difference. So be thinking about what kind of domain you might see or motif you might see with, depending on the purpose of the protein. Does it just need to bind DNA of all kinds? Does it need to bind a specific sequence? Does it need to bind RNA? So these are the kinds of things I want you to think about in terms of the different structures of these motifs and where you might see them in binding proteins. There's also the homeodomain, a little bit bigger than the zinc finger. About 60 very highly conserved amino acid residues. This is identified in proteins that function as transcriptional regulators, especially during eukaryotic development, which we're going to talk about that a little bit at the end of this chapter. The homeobox is the DNA sequence that encodes this domain. Then we have RNA recognition motifs. These are bigger still, 90 to 100 amino acid residues, and they're arranged in 4-strand anti-parallel beta sheets, and they're sandwiched against 2 alpha helices. I'll show you what that looks like in a second. This is found in some eukaryotic gene activators, where they might have to bind to DNA and RNA to induce transcription. So think about what kind of protein might have to bind both DNA and RNA. There are proteins that have these motifs that might bind to messenger RNA, ribosomal RNA, or other non-coding RNAs. And the long non-coding RNAs oftentimes compete with DNA binding and decrease gene transcription. This is something that we'll get back around to when we're talking about eukaryotes specifically. So these are examples of RNA recognition motifs. NF-kappa B is a regulator that we're going to talk about later on. But there are plenty of others. Pamilio, ROQ domain. There's lots of different proteins that are involved in recognizing RNAs and suppressing translation, or they could be, holding on to the RNA until the right time, and then that RNA will go and silence something. There's lots of reasons why you might want to sequester RNA. So regulatory proteins also have protein-protein interaction domains because they usually interact with the DNA and with some other protein. They either help recruit a protein or they bind to a protein and then recruit something else. So they have modular regions that interact with DNA, with protein, etc. So one example would be domains that help transcription factors dimerize that are necessary for the beginning of transcription. So let's talk about some of those domains. The leucine zipper is a very common one. It's got a lot of leucines in it, hence the leucine zipper. It was initially thought that, the structure literally had the leucines kind of like interweaving, but it's not exactly the case. But anyhow, this is an amphipathic alpha helix with a series of hydrophobic amino acid residues on one side and a hydrophobic surface, and then you've got polypeptides of a dimer. So we literally have two polypeptides coming together and interacting with each other. There's a leucine that occurs at every 7th position along these alpha helices, and they form a coiled coil. So think about a coil kind of coiling around itself. And it's found in a lot of different proteins. It's found in proteins with a separate DNA binding domain, but you can also see it in other proteins too. So this is an example of different regulatory proteins in eukaryotes. We've got some mammalian ones and then a yeast one. And you can see that there's a DNA binding region here that is rich in arginines and lysines. Then we've got a connector region, which varies drastically, but it's just a spacer. Then you've got the leucine zipper portion, where you see these leucines are conserved and their spacing is conserved. Below, we're looking at the crystal structure of one of these regions, okay? One of these proteins. And the region I want you to look at most closely is the zipper region. This is the coiled coil. These leucines are interacting with each other in such a way that brings them together. There's also the basic helix loop helix, and that's a conserved 50 amino acid residue stretch, and it can form two short amphipathic alpha helices. And there's a loop that links the two of them, which varies in length. These helix loop helix motifs can interact to form dimers, just like the leucine zippers can. This occurs in some eukaryotic regulatory proteins, and the majority of when you see that is when they're involved in development. So again, we're looking at two different proteins that are dimerizing. So they're coming together and forming, there's one protein here is donating a helix, another protein is donating the other helix. So this, the white and the purple, is showing their interface, the dimerization interface. In terms of eukaryotic regulation, like I said earlier, we have a lot of activators. We don't really do the repressor thing too much, but activators, absolutely. Yeast have 300 transcription factors for thousands of genes. Okay, so we're talking about, we've got to have some kind of regulation here, right? Lots of protein-protein interactions, and there's lots of overlap with a lot of genes being regulated by similar proteins. So most genes are subjected to regulation by multiple transcription factors. And what this is called is combinatorial control. It's a mechanism where you have mixing and matching of protein family variants to form dimers that are active as transcription factors. And using the different flavors of transcription factors and pairing them together gives you a more efficient and responsive way of regulating transcription. So now we're going to have a little bit of a departure. So I know this is chapter 28. However, we're going to be talking about chromosomes, chromatin, and we didn't cover chapter 24. So I just want to give you a little bit of background on that, just so that you're not completely lost. You don't need to memorize this information, but it will be incredibly helpful for what is to come. So chromatin is the eukaryotic chromosomal material. and it's made up of DNA, RNA, and proteins. It's all compacted around proteins, DNA, and stuff. It's just wrapped up very tightly. It kind of looks amorphous in G0 and in interphase. But in S phase, you have replication. And so it looks a little bit different. So if you don't remember mitosis and kind of what that is and the different states of the cell, G0 versus G1, S, G2, then you might want to refresh yourself with your biology. We're not going to go over, prophase and metaphase and all that stuff. But just kind of remember what is S phase, just kind of the very basics. So the DNA gets packaged around proteins that are called histones. And those histones are proteins that tightly associate with the DNA to make it one big package. So you take some DNA, you wrap it around the histone core, which there are several flavors of histones that we'll talk about in a second. You wrap around the DNA, and then you form what's called a nucleosome. Between each nucleosome, there's a bit of linker DNA. This helps to compact all the DNA in your cells. You can actually see them. These very dark bits, those are nucleosomes. But what are histones? We said they're proteins. They're small, and they have a lot of basic residues. There are 5 major classes of histones, H3 and H4. So they're designated by H for histone and then a number. H3 and H4 are nearly identical in all eukaryotes. But H1, 2A, and 2B have less sequence similarity. But what you can see is that they have a lot of basic amino acids. Chromosomes are arranged as what's called the beads on a string. And those beads are complexes of histones and DNA. So each nucleosome has 8 histone molecules, 2 each of H2A, 2B, H3, and H4. And each nucleosome contains about 200 base pairs of DNA, the majority of which is tightly wound around the core, and then there's that little bit of linker, like I said. That linker leads you to the next bead. This is showing you how DNA wraps around a histone core. This first part is just the histone core itself. The histones interact with each other. And again, you're not required to know that H4 and H3 make a dimer or anything like that. I'm just showing you so that when we talk about chromosome remodeling and chromatin remodeling proteins, you'll have a better idea of what that looks like. So now add on the DNA. You wind it around, and this is what it looks like. So there's the space filling model here, and then there's the ribbon diagram with showing the individual nucleotides and their pairing. But that's what a nucleosome looks like. Your chromatin is just long stretches of this. Now, something else very special about these histones is that they have these extended amino terminal tails. So remember, that's the N terminus. The very beginning. They're intrinsically disordered, which means that they don't really have a structure. They're just kind of floating out there. This is where histone modifications occur. And these modifications that happen are key in forming contacts between nucleosomes and chromatin. So depending on the state of the different tails, the amino terminal tails, that will govern partially how tightly compacted your chromatin is. So histone binding itself is actually not random. The binding of the histone core seems to be localized to places where there are lots of AT base pairs. Again, you do not need to know this, just for your understanding so that it doesn't come as a surprise. So now that we have a little bit more information about chromatin, we can skip over 28.2 about bacteria and go right to expression in eukaryotes. We got to talk about transcriptional ground states. What that means is the inherent activity of a promoter and all the transcriptional machinery in vivo. In bacteria, RNA polymerase can pretty much initiate transcription at some level on everything, everywhere. But in eukaryotes, strong promoters are generally inactive without regulatory proteins. So that's a bit of a departure. We are different after all. So access to eukaryotic promoters is restricted because of the structure of chromatin, which is why I inserted that. You got to have that in your mind. All this DNA is wrapped around these histone cores to make nucleosomes. You can't really get in there and bind and, make an mRNA transcript. Usually you have to have some chromatin remodeling. Positive regulation mechanisms are more prominent than in bacteria, and they are required for transcription. So remember, positive regulation, think activators. And there's lots of regulation involving those long non-coding RNAs. Eukaryotes have larger, more complex regulatory proteins, and transcription occurs in the nucleus and translation occurs in the cytoplasm, so that it's separated in space and time. These are all features of eukaryotic gene regulation that you don't see in bacteria. Transcriptionally active chromatin is structurally distinct from inactive chromatin. We talked very briefly about the architectural proteins that can bind and help DNA to loop. Well, we're going to be talking about those kinds of things only on steroids here. There's heterochromatin, which is more condensed, and it is transcriptionally inactive. Approximately 10% of chromatin in a typical eukaryotic cell exists as heterochromatin. And this is usually associated with the structural parts of the chromosome, so like the centromeres. You don't need to access that, so stay off of it. I'm going to be condensed. Leave me alone. However, euchromatin is less condensed, and you can visually see the difference with microscopy. Because of the existence of nucleosomes and DNA being tightly wound around these histone cores, you have to have some remodeling. And that's again called chromatin remodeling, which is transcription-associated structural changes in the chromatin. What happens when you remodel is you reposition the nucleosomes, so you maybe slide them down or kick them out altogether. You might replace some of the standard histones with a histone variant that maybe doesn't bind DNA quite as tightly. And there can also be covalent modification of the nucleosomes. Remember, those tails are hanging out, waiting for modification from the histones. This table tells you all the different enzyme complexes that can catalyze chromatin remodeling. And we're going to talk about kind of the major ones here. So this is a good summary table. We'll start with the SWI/SNF and the I-SWI complexes. So SWI/SNF complexes remodel chromatin so that nucleosomes are ejected near transcription start sites. And yeah, you heard me right, ejected, kicked out, booted out. You're evicted, nucleosome. So that histone core, boom, you're out of here. It's involved in a cycle that replaces the nucleosomes with transcription factors. Then we have the I-SWI complexes, which optimize nucleosome spacing so that the chromatin assembly and transcriptional silencing can happen. So we've got to be able to, fix what we broke, right? So when you have to do transcription in a particular area, a transcription factor will replace the nucleosome, and this requires ATP. When you don't need transcription anymore, then the nucleosome will come back and bind, and the transcription factor gone. So with these SWI-SNF complexes, you have to have ATP. This is why regulation is expensive. If we just had all of our promoters out there ready to be accessed, we wouldn't have to be spending ATP just to make the promoters accessible. But this is how we do it. The SWI-SNF complex will bind, and then the transcription factor will come and replace that whole complex, and in turn, you've kicked out that nucleosome, that histone core. Instead, you have the transcription factor bound, and then you can start doing all of the transcription, and then replace that transcription factor with your nucleosome core when it's time. There are other families that do chromatin remodeling. So the CHD complexes, they do a mixture of things. They might eject the nucleosome to activate transcription, or it'll do the reverse and assemble chromatin to repress transcription. So kind of like with enzymes where they can do the forward and reverse reaction, but the contents of the cell and all of the things influence which reaction is more favorable. It's kind of like that here, where you have some complexes that will eject the nucleosome, some that will reassemble it so that you can repress transcription. And you need both of these activities. And one way that you can modulate that is to control how much of each complex you have, so that you have kind of the push and pull, and overall you have the regulation of whatever gene you're trying to regulate. Then we have the NO80 complexes, and these remodel chromatin for transcriptional activation and for DNA repair. So remember that DNA repair is always ongoing, and in order to have access to the DNA, you have to kick out a nucleosome. So SWR1 is a subunit. It promotes the subunit exchange in the nucleosomes to add in histone variants. So we talked about the generic flavors, but there are variants of these histones that you can swap in and out that will change how they interact, how the whole histone core interacts with the DNA. Finally, you can have covalent modification of histones. So the core histones are modified by methylation of lysine or arginine residues. Phosphorylation is also a big one. Acetylation, ubiquitination, sumoylation, which sumo is essentially like a smaller ubiquitin. And all of these sites are located on that wiggly wobbly N-terminal domain. So different patterns of modification indicate that there may be some kind of a histone code. Have researchers discovered what it is yet? Not really. But there are certain modifications or certain combinations of modifications that are generally associated with increasing transcription or repressing transcription. But it's not, set in stone, this is what it is. And looking at different nucleosomes, you may have the same set of modifications, but, on may look different from nucleosome 1 to nucleosome 20. So there's plenty of caveats there. Some of the enzymes involved in this are histone acetyltransferases, also called HATs, and they will transfer an acetyl group to a lysine. Pretty simple. There are also methylases that will methylate lysines as well. And you could imagine that since a lot of this is happening on lysines and arginines, and there's lots of enzymes, that they're all competing for the same spots. That in and of itself is regulation. But these hats will acetylate particular lysine residues, and there are two different types of hats. There's type B, which will acetylate newly synthesized histones. So that's part of the whole protein metabolism, right? And then there's type A, which will acetylate nucleosomal histones following H3 methylation. So it recognizes methylation on that N-terminal tail, and then it says, okay, my turn, and it'll add an acetyl group. Lysine acetylation decreases histone affinity for DNA, and that results in an increase in activation, so you can transcribe your message a little bit more readily. Think about what it is about acetylation that would decrease the affinity for DNA. Now, because we can add on an acetyl group, we have to have a way to take it off. And these are histone deacetylases. These are enzymes that take off that acetyl group. Now, there's been a lot of, I'm not going to say argument, but conversation about what these enzymes should be called, because, they're called histone deacetylases, but they actually work on acetylated lysines, and they can work on things outside of the histones. So science is always ongoing, right? There's always more to do. But these enzymes typically mark chromatin as transcriptionally inactive. There are lots of different flavors of histone deacetylases. There's different families. We're not going to go into all that. We're going to pause here and say this is it for video 1. We'll pick up with the remainder of section 3 in chapter 28 in the second video, where we'll finish covering all of the nitty-gritty details about gene regulation in eukaryotes. Until then, please stay safe and make sure that you tune in for the next video.